So whenever a host is infected by a virus, so the first thing that happens is that the proteins that are present on the surface of the virus will stimulate the production of antibodies. And these antibodies are basically stimulated by or produced by a host to protect the host from the viral infection, wherein it is going to kill the virus and stop the infection. But in some cases, you can have the viruses which will escape the antibodies, mainly because the proteins that are there on the surface of the virus, especially the spike proteins that are there's a high chance that the spike proteins can have large number of mutations in them. So the genes that code for them have the ability to mutate, and because of which you can have an altered spike protein. So suppose this is the spike protein that was there initially, later on you can have a spike protein something like this. So if antibodies are generated for this spike protein, obviously they will not be effective, or they will not be able to react with this particular spike protein. One classic example of this is an influenza virus, which goes on changing its spike protein, and that is one of the most important reason why you cannot get, sorry, I mean to say you can get influenza or common cold more than once. This, I guess, everyone would be familiar with, because in a year, maybe once or twice, we do get affected by influenza. And again, in today's scenario, if you will see, in case of COVID-19 also, we have predictions that there are certain mutations which are taking place on the spike protein, because of which you can have a high chance of reinfection also. Coming to the general morphology, so as I said, you can classify viruses based on nucleic acid. You can also classify viruses based on the morphological types. Like, for example, you have helical viruses, wherein the virus is a long rod, which can be either rigid or it can be flexible, and basically it has got a cylindrical capsid, uh, which is there, and inside that you have your nucleic acid. So this is the capsid, this whole thing is capsid, which is cylindrical, and these are the capsomeres inside which you have the nucleic acid. The example of viruses with the helical structure is the rabies and the Ebola virus. So here in this pic, micrograph of the Ebola virus is shown. As you can see, this is a helical rod-like structure that is seen. Then you have polyhedral viruses. In this, you have many animal, plant, and bacterial viruses which come into this category. So basically, a polyhedral virus will have a shape of a icosahedron, which is nothing but made up of 20 triangular faces and 12 corners. So these are the triangular faces that are there. So you'll have 20 of them, and you will have 12 corners. And each of these triangular faces that are there, they are an equilateral triangle. An example of a polyhedral virus is the adenovirus, as well as the poliovirus. Then you have something called as an enveloped virus. So as we have already seen, some of the capsids can also have an additional covering on them, which is called as an envelope. And mostly the envelope viruses are roughly spherical in shape. This is an example of an influenza virus, which is an enveloped virus, and which has got a spherical shape. But you can have, if you can have helical as well as polyhedral viruses which can have envelope. So if it is a helical virus which has got an envelope, it is called as an enveloped helical virus, or a enveloped polyhedral virus, which is the case of influenza. Here again is another example of herpes virus, which is an enveloped virus which has got an icosahedral uh, structure, capsid, and on top of that you have got the envelope that is there. Then the last category is for complex viruses, wherein the structure can be very complicated. It can be a mixture of many things. For example, is your bacteriophages, that is the viruses that infect bacteria. So some of the bacteriophages have got capsids, and they have got additional attachment to the capsids. For example, this is for a T-even bacteriophage, wherein you can see this is the capsid which is there, which is a polyhedral structure inside of which you have your nucleic acid, and it has got an additional sheath kind of a thing which is a tail, and this is helical, and you have tail fibers that are attached to the sheath, and you have pin-like structures. Basically, these are used for, fibers are used for attachment, and these pin-like structures are also used for attachment. And an example of this complex virus is your pox virus, that is of the category of orthopox virus, which you can see over here, wherein the structure is not quite clear in case of what type of capsid it has got, but then you can see several coatings that are there which surrounds the nucleic acids. So how do you name viruses? So the genus ends with a virus. So like if you have herpes virus, you can call it as herpes virus or a simplex virus. The family name ends with a viridae, so you have herpes viridae, and the order ends with ales.